In this lab, we will be analyzing a variety of lipids. So what are lipids? These are a diverse class of biomolecules that are very hydrophobic or very slightly polar. They are generally insoluble in water to slightly soluble in water. They are soluble in organic solvents and have hydrocarbon chains. In this lab, we will be studying the chemical nature of these three types of lipids. Steroids, they contain a 3 6-membered ring and 1 5-membered ring. Cholesterol is a prototypical member of steroid lipid and is produced by the liver of animals. Triacylglycerols contain a 3-carbon backbone that's composed of glycerol that's attached to 3 fatty acids by an ester bond. It is the most abundant form of lipid in the body and is a stored of chemical energy. And then phospholipids are similar to triacylglycerols in that it has a 3-carbon glycerol backbone, but instead of being attached to 3 fatty acids, it's actually attached to 2 fatty acids by an ester bond. And then the third carbon is attached to a phosphate group and then to another group, which is a short chain organic group. And phospholipids are abundant in membranes of cells. In part A of this lab, we will extract lipids from nutmeg and egg yolk extracts. Nutmeg is abundant in a lipid called trimyristin, which is shown here. This is a triacylglycerol. And egg yolks contain a wide variety of lipids, but is particularly abundant in cholesterol. In part B, we will analyze various lipid samples. So in addition to the nutmeg and egg yolk extracts that we create, we will also analyze lecithin, which is a lipid from vegetable sources, a vegetable oil, and butter. And we will perform 3 different tests on these various lipids for the presence of phospholipids, triacylglycerol, and cholesterol. So the phospholipid test is going to test for the presence of phospholipid. It's confirmed by this blue color generated when a dissolved sample is spotted on a piece of filter paper and treated with molybdate, which is a staining agent. The hydroxymic acid test detects the presence of an ester. The development of a burgundy to purple color in a sample will indicate the presence of an ester. And triacylglycerols and phospholipids will test positive in a hydroxymic acid test. And then the last test that we will do is the Lieberman-Burchard test. This tests for the presence of cholesterol. If cholesterol is present in the sample, it will turn a greenish blue color upon addition of acetic anhydride and concentrated sulfuric acid.